Yeah, as you guys heard, I'm going to talk about, sort of themes related to excitatory-inhibitory balance and related to inhibitory circuits and how they relate to psychiatric disorders. And I'm going to divide my talk into two parts. In the first part, I'm going to talk about a paper that we published last year on gamma oscillations and their role in cognition that's relevant to schizophrenia. And in the second part, I'm going to talk about mouse models of autism, social behavior, and multi-neuron encoding. And I'll take a break in between. So I'll answer questions about the first part about halfway through my talk. All right, so, why am I talking about gamma oscillations and cognition? When I say schizophrenia, probably the first thing that pops into most people's minds is, hearing voices or paranoid delusions. But we now know that deficits in cognition are actually the core of schizophrenia. And, what we consider higher order cognition, executive functions, things that involve a lot of planning and organization, those are the abilities that are usually the most affected, and they're sort of quintessentially mediated by the prefrontal cortex. Now, one of the tasks that best exemplifies this and that's most impaired in individuals with schizophrenia is the Wisconsin card sorting task. So just to give you a sense of what the cognitive deficits are like, let's talk about this task. Some of you are probably familiar with it, right? In this task, you would get like one card here with two red crosses on it, and you'd have to sort it into one of these four piles, but you wouldn't be told the rule for sorting, right? So you could sort based off of color or based off of shape or the number of objects. And after each trial, you're told whether you do this right or wrong. And pretty soon you figure out the rule. And then without warning, the rule will change. And you have to realize that it's changed and figure out what the new rule is. Now, this is obviously a complicated human task, and we can't perfectly model it in rodents, but there's certain aspects of it that we can try to capture. And in particular, I'll note, this isn't like, it's not an intentional or perceptual task. It's not hard to detect the stimuli and figure out what's on each card. The hard part is to realize when rules have changed. And furthermore, when rules change, you have to start paying attention to information that was previously irrelevant, right? So if you're initially sorting based off of color, you're probably just glossing over the shapes of the objects. And when the rule changes, you have to start paying attention to something that was previously irrelevant. So, we have a mouse task that captures those key elements of the task. In this task, mice are presented with like two bowls on each trial, each one with different odor and texture cues. And one of these four cues, in this case an odor, will tell the mouse where a hidden food reward is. The mouse will learn this initial association, and then without warning, the rule will change. And it can change in a couple of different ways, right? We can do an intradimensional rule reversal, where like odors would still stay relevant, or we can do an extra-dimensional rule shift, in which we would switch from an odor-based rule to a texture-based one, or vice versa. And the key thing about a rule shift is that without warning, the mouse has to start paying attention to cues like textures that it was probably ignoring before because they were irrelevant to the task. Okay, so I mentioned that we're interested in the role of gamma oscillations in this task. Many of you're probably familiar with gamma oscillations, right? If you record electrical activity from the brain in almost any way, you can see oscillations. And these oscillations in the higher frequency bands between about 30 and 100 hertz or quintessentially around 40 hertz are called gamma oscillations. And they're recruited during many cognitive tasks. So when humans do tasks that involve cognitive control and behavioral flexibility, you often see gamma oscillations appearing and increasing in power and synchrony over the frontal cortex. Now, a lot of work, including the work I did as a postdoc, has shown that parvalbumin interneurons often play key roles in generating these gamma oscillations. And studies of individuals with schizophrenia have shown that, when you look at brain tissue after these individuals have died, there's something wrong with their parvalbumin interneurons, particularly in the prefrontal cortex, right? And so you might reason that perhaps gamma oscillations would be abnormal in schizophrenia, and indeed that's the case. Many studies using many paradigms from many labs, and you'll hear about some of those, I think, from Peter Uhlhaus later, have shown that when individuals with schizophrenia do cognitive tasks, there's often deficiencies in the gamma oscillations that are recruited. So you can start to put all this together and imagine like, hey, maybe these problems with PV interneurons cause problems with gamma oscillations that cause problems with cognition. All right, and this is the basic controversy that we want to get at today. I think for decades, there's been a huge debate in neuroscience about the significance and meaning of oscillations, right? Many people rightfully point out that, anytime you have excitatory and inhibitory neurons coupled, it's a feedback loop and it's going to generate oscillations. And based off the time constants of synaptic inhibition, these oscillations are usually in the gamma frequency range. So these oscillations are just epiphenomena. And even when you see them synchronized between neurons in different locations, it probably doesn't mean anything. It's just a sign that these regions are connected. And it's just maybe these synchrony that you see is just volume conducted across regions, and it's not functionally meaningful. And this is really important in the context of schizophrenia, right? I don't think there's any question that gamma oscillations are a biomarker for something being wrong with the underlying circuits. But the question that we are going to address today is whether they're actually a key mechanism that should be targeted to restore cognition, right? It's not just that they're markers of circuits being active, they are actually the thing which makes the circuits work correctly, okay? So we wanted to really get at the heart of these controversies. And so one thing we wanted to do is we wanted to get around these issues with electrical recording. So sometimes people see synchronization of activity at two different sites, and other people say like, maybe that's not real. It's just due to some artifact that's being conducted between them. So to get around this, we took advantage of voltage indicators. So we measured fluorescence from individual cells to measure their voltage. And so this image here shows a reference fluorophore, TD tomato, and a genetically encoded voltage indicator, ACE-M neon, which is in green, in parvalbumin interneurons. And so we can measure fluorescent signals from parvalbumin interneurons in the left and the right prefrontal cortex, and we could measure the synchrony between them. Now, I won't go into the details of how we did this, but very briefly, we basically filtered the left and the right prefrontal cortical signals in this gamma frequency range, and we looked at how correlated they were at each moment in time, and we compared that to correlations we observed in shuffled data, okay? And so we could see how often the real correlations at gamma frequency exceeded the correlations that we would expect based on chance, and that would be our metric for synchrony, okay? So here we basically plotted how often we're seeing the appearance of statistically significant synchrony at different times. And this is the key finding right here, which is that right after animals make an error during rule shifts, we see this increase in gamma frequency synchrony, okay? Now, this is really interesting because remember, mice are not told when a rule shift happens, right? They have to infer this. And so basically they're going along, they're following the rule that they learned during the initial association, they're getting food reward, and then all of a sudden they follow that rule and they don't get an expected food reward. So that error, that time when they don't get the reward they expect, that's a signal to them that, hey, this rule has become outdated and maybe it needs to change. And right at that moment is when we see this increase in gamma synchrony following incorrect trials relative to baseline. This increase in synchrony doesn't happen on correct trials. And furthermore, it's specific for frequencies around 40 hertz. So we don't see it at higher or lower frequencies, okay? So here we see that in normal mice, when they're doing this rule shifting task, there is this synchrony that appears between parvalbumin interneurons in two different locations, the left and the right hemispheres, specifically within the prefrontal cortex. And so we've been able to measure this using voltage indicators and confirm that it's real. Our next question is, does it do anything? Does it matter for normal cognition? So to answer this question, we decided to use optogenetics, right? And we expressed channel rhodopsin within parvalbumin interneurons in both the left and the right prefrontal cortex. And then we can deliver light flashes to stimulate these two sets of interneurons. And we can deliver those light flashes at different frequencies and with different phase relationships. And in particular, we can deliver very weak light flashes at 40 hertz, but do so out of phase between the hemispheres, so as to disrupt the normal in-phase synchronization that we observe following rule shift errors, okay? So when we do that, what we see, and we measure rule shifting performance by the number of trials it takes mice to learn a rule shift. What we see is that in mice that have channel rhodopsin, when we deliver this out-of-phase stimulation, it becomes really hard for them to learn a rule shift. They take much longer to learn the rule shift. And in fact, if we look at what they're doing during that time, they're very perseverative. They're stuck on the initial rule and they're unable to switch normally. We can deliver stimulation at other frequencies, like at 20 hertz. We can deliver 40 hertz out of phase or 40 hertz in phase stimulation. We've done this a lot of different ways. And what we always see is that it's only out of phase stimulation at 40 hertz that disrupts performance. Out of phase stimulation at other frequencies like 20 hertz has no effect. And in phase stimulation at 40 hertz doesn't make their performance worse, and actually maybe makes their performance a little bit better. Okay. Now, how is all this relevant to schizophrenia, right? I kind of mentioned that this task may be relevant to the sorts of cognitive deficits that we see in schizophrenia. And in fact, we've studied mice which model aspects of schizophrenia. These are mice that have mutations in two transcription factors, DLX5 and DLX6, that are necessary for the normal development of parvalbumin interneurons. And in a previous study, we showed that these DLX5/6 mutant mice, they have abnormal parvalbumin interneuron physiology. And so that sort of models the deficits that are seen in the brain in schizophrenia in parvalbumin interneurons. They have deficits in this rule shifting task. So they are very perseverative. They learn the initial rule just fine, but then they get stuck on it. And that kind of resembles a lot of cognitive deficits, for example, in the Wisconsin card sorting task that are seen in schizophrenia. And just as in schizophrenia, if you measure gamma oscillations, you see that they normally increase during these sorts of tasks, but they're deficient in these mutant mice. And what's interesting is that all of these deficits appear following puberty. And that very much resembles what you see in humans, where the typical age of onset of schizophrenia is a sort of like post-adolescent, early adulthood period. Okay, so what happens in these mutant mice? Well, I've already told you what happens in normal wild-type mice, and that's shown here in gray. I've told you that in those mice, if we measure synchrony, specifically in the gamma frequency range, while mice are doing this rule shift task, we see this increase in synchrony on incorrect trials relative to correct trials, okay? And that's what's plotted here, that synchrony is higher on incorrect trials than on correct trials across animals, and this doesn't happen at lower or higher frequencies. If we now look at mutant mice, shown here in blue, then as we sort of expect based off of our earlier work where we recorded electrically, they don't increase gamma synchrony, okay? So this normal increase in 40 hertz synchrony that occurs on incorrect trials relative to correct trials is absent in these mice, okay? And so now we can try delivering different patterns of optogenetic stimulation. We can deliver stimulation to parvalbumin interneurons that's at 20 hertz. We can deliver 40 hertz stimulation that's either out of phase or in phase. And we can measure how many trials it takes these mice to learn. And what we see is that the 20 hertz stimulation doesn't improve their performance. 40 hertz out of phase stimulation doesn't improve their performance, but their performance improves markedly. And actually they perform just like wild type mice if we deliver 40 hertz in phase stimulation. So just to summarize, normally we see this 40 hertz synchrony appear in parvalbumin interneurons during rule shifts. We can optogenetically disrupt that synchrony in normal mice and make them perseverative, or we can take mutant mice that are perseverative and restore this synchrony, and we enable them to learn normally. Something that's quite interesting and quite fascinating to wonder about is that these pro-cognitive effects of 40 hertz stimulation are persistent. So once we rescue the performance of these mice, it stays good for days, weeks, even like a month. Later, we can just test them again without delivering any additional stimulation, and these mice will continue to perform normally. So they've really learned something and had kind of a eureka moment and undergone some kind of circuit plasticity as a result of this manipulation. All right, just a few other things to note. Everything I've told you is cell type specific. So if you measure voltage synchrony from somatostatin interneurons, which are different subtype of interneurons, you don't see increases in gamma synchrony. If you deliver out of phase stimulation to somatostatin interneurons, it does not disrupt rule shifts. And if you stimulate somatostatin interneurons in phase in these mutant mice, that does not improve their rule shifting. Okay, so everything I've told you is only true for parvalbumin interneurons. It's also only true for rule shifts. So there's something about this process of starting to pay attention to cues that are previously irrelevant that depends on gamma oscillations. You don't see increases in PV interneuron gamma synchrony when mice learn an initial rule or when they do an intradimensional reversal of that rule. Out of phase stimulation doesn't disrupt initial learning or just like reversing a rule from one odor to another odor. And these mutant mice are not impaired when they learn an initial rule or when they do rule reversals. So just to summarize this first part, and then I'll pause for questions. These increases in gamma synchrony are real, right? People often speculate, oh, these electrical signals may be contaminated by artifacts, which create synchrony. No, we've done measurements of voltage signals using fluorescent indicators, and we still see the appearance of the synchrony, and it happens at a key time during behavior. Furthermore, it's actually essential, right, for rule shifting and for this aspect of cognition. We can causally manipulate synchrony, and disrupting it impairs mice, whereas restoring it in mutant mice enhances their cognition. And this role is cell type specific, frequency specific, and it's specific for these rule shifts, okay? And then finally, not only does this resolve kind of the questions about, is gamma synchrony really important? It shows that not only is it important, it might have very powerful therapeutic effects. And if we target the right cell types at the right frequencies and with the right phase relationships, we can have very powerful restorative effects on cognition that might be relevant to restoring cognition in conditions such as schizophrenia. Okay, so I'll go ahead and pause there and see if there's any initial questions.
Maybe I can have the first question myself. So I was wondering, now you're talking about synchronization left versus right side. I was wondering about the synchronization within one of the sides. So with your laser, you will synchronize all the neurons on one side. Would that have any effect?
Yeah, so I think, we're taking advantage of the fact that when we do either optogenetics or these measurements of voltage indicators, right, we don't have single cell resolution. And so a convenient way to look at synchrony is through this lens of left hemisphere versus right hemisphere, right? It's easy to separately manipulate and separately measure those two. Whereas if we do things, the prefrontal cortex in mouse is very small. So obviously, if we try to do things within the same hemisphere and we stimulate at one site, it's probably going to bleed over into, a neighboring site a few hundred microns away. So certainly, if we look at electrical signals within a hemisphere, we do see that they tend to increase and decrease, suggesting that these increases in cross-hemispheric synchronization are also happening locally within a hemisphere. But we kind of haven't experimentally gotten at that because we're just taking advantage of this fact that there are two populations that are separated by some distance in the two hemispheres.
Right. So we don't have any questions right now, so
okay.
Please, the audience, if you ask, but I have another question. So I was wondering, so now you show you can improve this particular behavior. So does this also generalize? So if you look at the regular behavioral repertoire of those mutant mice and how they differ from the controls, does anything else change as well as a consequence of this sort of training?
Yeah, this is a good question. I mean, I think we don't know fully the answer to that question. And part of the issue is that, in order to see these kinds of effects, these mutant mice have a very specific cognitive deficit. They're impaired on rule shifts, but not on learning of initial associations or rule reversals. So we think there's a very specific aspect of cognition that is disrupted here. And it's a little bit difficult for us to find other, these are not mice that, unlike a lot of other mutant mice, have wide-ranging cognitive deficits, right? And I think that's important. I think if they did, it probably is harder to rescue their cognition. And so the short answer to that is we don't know yet. It's a very interesting question that we're interested in for sure. I can also go on and we can do all the questions at the end if that seems easier.
Well, now we're getting now
okay.
questions. So the first one is, did you always use exactly 40 Hertz, or if you were to use 39 or 41 Hertz?
This is a good question. We've also looked at this effect using 60 Hertz stimulation, at least in the mutant mice. We've shown that 60 Hertz stimulation also rescues their behavior. So I don't think it's exactly 40 Hertz. I don't think it's an infinitely broad range either. I don't think goes all the way down to 20 Hertz, but I think within this sort of gamma frequency range of maybe 40 to 60 Hertz, there's some flexibility.
Yeah, right. And the next one would be, are pyramidal cells synchronous as well during gamma synchrony between PV interneurons?
Yeah, this is a good question. So what we know so far is that certainly when you deliver even weak optogenetic stimulation, it very quickly entrains the PV cells. And when you entrain the PV cells, it entrains the pyramidal cells. And that's shown in a supplementary figure in this paper. What we don't know is during normal behavior, how strongly are the pyramidal cells entrained. And what we think is that they are entrained, but different subpopulations of pyramidal cells are entrained differently. So that's something that we're in the process of looking at right now. Yeah, it's a great question.
Yes, and sometimes people talk about the ing or the ping model.
Right. We don't know the answer here, but we are also trying to perturb specific sets of synapses or specific populations of cells and see what happens to the overall synchrony. And so I think that these are the questions that I think I have a postdoc who's working on these questions right now, and we hopefully will have an answer in a couple of years. And certainly if other people are interested in working on it, feel free to get in touch.
Right. Yeah, so maybe do you think we have time for one more before
we could maybe do one more and then I'll go on, yeah.
Yeah, okay, in REM sleep, we have gamma oscillations as well. Can we connect such positive effects on cognition to REM sleep?
Yeah, it's a great question. Like, is gamma, here we've shown a very specific role for gamma in one particular context, and maybe gamma, when it happens in other contexts, is doing something similar. Certainly, it seems like what's happening here is that the gamma oscillations are not just important for the immediate performance of the task, but they're rather important for learning something about task structure, because the effects of potentiating gamma are long lasting. And so it's interesting to speculate that when you have gamma during REM sleep, it may similarly lead to like learning of new kinds of circuit representations or some kind of circuit plasticity that has effects later on during wakefulness. All right, so thank you for those excellent questions. I will go on now to the second part of the talk. I'm going to talk about this work that was led by Nick Frost, thinking about EI imbalance in autism and social behavior. So, you know, there are a lot of mouse models of autism, and one of them that's the most studied are mice which lack SHANK3. This is a gene that is strongly associated with autism. Humans who lack this gene often have a syndrome, which includes autism as an important component. And many labs have shown that mice that lack SHANK3 have social deficits. So this is just our version of that. This is how much time wild type or SHANK3 knockout mice spend interacting with a novel mouse, and you see that the knockout mice spend less time interacting. We're of course interested in like what's happening in the brain during this that might be contributing to this or related to it. So we did microendoscopic calcium imaging, right, where we implant a Grin lens into the prefrontal cortex, and we measure GCAMP signals from something like 50 to 100 neurons at once. And here's an example of 10 traces when the mouse is either alone in its home cage or when it's socializing. And in this case, the mouse socializes with one new mouse, then a second new mouse, then the first mouse again, and then the second mouse again. And you can see some cells increase or decrease their activity during these periods of socialization. And so what you can immediately notice is that in wild type mice, right, roughly the same number of neurons will increase or decrease their activity during socialization. In SHANK3 knockout mice, something a little counterintuitive happens, which is you still have neurons which increase their activity during socialization, but you actually have too many neurons increasing their activity during socialization. And I think naively, that's not what you might expect. You might expect that, you know, not enough neurons were turning on, and that's why these mice were not interested in socialization. But no, actually too many neurons turn on. And kind of the question we want to answer is, why is this a bad thing? Why is it a bad thing to have too many neurons that seem to like socialization? Okay. And so I'm going to take a long meandering route to answering that. And basically what we want to do is first talk about how can we decode the neural activity that's related to socialization, right? So this is a common thing that people do now in systems neuroscience. We get these big data sets, and so we say like, hey, let's take our big data set and try to decode something about behavior. So you take your neural activity, you feed it into some kind of classifier, and you see if you can classify when the mouse was doing thing A versus thing B, like alone in its home cage versus socializing with a mouse, or socializing with mouse number 1 versus socializing with mouse number 2. Now for this, we're going to use a neural network classifier, and I'm going to explain first a simple idea for why to do this, and then a more complicated reason why this turns out to be a good idea. You know, simplistically, there's a lot of different choices of classifiers you can use. And we thought, like, why not use one that reflects what the brain does? So imagine you're a downstream population of neurons and you're getting input from the prefrontal cortex. Can you use that input to predict whether the mouse is engaged in socialization or not, right? And so we have this very simple linear 3-layer neural network. The input layer are real neurons. So each neuron in the real in our recording corresponds to one neuron in this input layer. These input neurons have fixed random connections to hidden layer units. The hidden layer units sum up all their inputs and then use modifiable output weights to propagate that activity to a single output unit, which then classifies what the mouse was doing, right? And so during training, right, we would take a frame, we would activate the corresponding neurons in the input layer, propagate their activity through the network, make a prediction about behavior, see if we were right or wrong, and then use a simple gradient descent algorithm to adjust these weights. I'm not going to go through the details. You can look at the paper for that. And then we'll test our network, right, by feeding in one frame at a time. Again, we activate the corresponding neurons in the input layer, propagate that activity, compute the activity of the output unit, and depending on whether it's low or high, make a prediction about whether the mouse was being social or whether it was alone in its home cage, right? So why do we do it this way? Why don't we do something much more standard and just use like a linear SVM or a logistic classifier? Okay. So the reason is because it makes a difference depending on how activity is being encoded in the brain. Okay, and so let me do a little bit of a deep dive into this. So usually we think about changes in levels of activity of individual neurons, right? So we might have two states, one corresponding to when the neuron is alone in its home cage and another corresponding to when it's being social. And so imagine we just create activity that differentiates these two states, right? So we have some activity raster that's associated with periods when the mouse is alone in its home cage. And we're going to create an artificial activity raster from this that represents periods of social interaction. And the way we do this is by changing individual neuron activity levels. So we take activity from neurons 1 through 50, and we transfer it to neurons 51 to 100. And so in our second raster, which represents periods of social interaction, basically neurons 1 through 50 have decreased activity, and neurons 51 through 100 now have increased activity. And so then we can compare activity during any one of these frames, and based off of the differences in the activity levels of the individual neurons, we can kind of infer whether this was probably a period of social interaction or a period when the mouse was alone in its home cage. In this type of situation, we can measure how well our neural network classifier performs, or how well a linear SVM, or it looks the same if you look at like a logistic classifier, how well they perform as a function of how much activity we move. So the different colors here represent shifting more and more activity and creating larger and larger differences in activity levels. And basically, obviously, the greater the activity level difference, the better the performance is. And the performance is almost exactly the same for a linear SVM and for our neural network classifier, right? It's actually very slightly better for the linear SVM. Okay, but this is not the only way that neurons can encode things, right? What if individual neurons don't change their activity levels in these two conditions, and all that changes is correlated activity or coactivity? So now we start with the same raster for when the mouse is alone in its home cage, and then we create artificial rasters for when the mouse is being social by taking this raster and just rearranging the activity of neurons 1 through 10. So the activity level of neurons 1 through 10 hasn't changed here, but we've just rearranged when they're active so that they're now active at the same times, creating patterns of correlated activity or coactivity. And we can create one pattern, or we can add a second pattern or third pattern. And now activity when the mouse is alone versus when it's socializing, the activity levels of individual neurons aren't different. All that's different are these patterns of coactivity. And now what we find is that a linear SVM has a really hard time with this. It really just performs at chance levels in distinguishing these two sets of rasters. But our neural network classifier, which it's a linear neural network, but because of the way the learning rule works, it learns to recognize these patterns of coactivity. And it either detects these periods when all the neurons are coactive, or else these periods when all the neurons, 1 in 10, are silent, and it's able to perform at above chance levels at distinguishing these two types of rasters, which differ only in coactivity, okay? So this is great. Now what we'd like to know is, hey, if we look at our real data, how is information being encoded? Is it being encoded just by changes in activity levels, or is there extra information that's available in coactivity? Okay, and one way we could answer that question is to train our classifier based off the real data, and then feed in data in which we preserve changes in activity levels, but we either remove or preserve the changes in coactivity. And if our classifier performs better when we preserve the changes in coactivity, then we know that the coactivity was transmitting additional information. If it doesn't matter if we preserve or get rid of the coactivity, then we know, this information and activity levels was enough. That was all the information that was present. So how can we do that? Well, we train our network based off of the real data, and then we feed in two types of shuffled data. In one case, we shuffle so that we preserve changes in activity levels, but we disrupt correlations. And we do that using what we call a swap shuffle. And in a swap shuffle, we make reciprocal swaps between neurons. So like here, we'll shift a little bit of activity from neuron A to neuron B, and then at a later time point during the same behavior, we'll shift activity from neuron B back to neuron A. And as a result, during this whole block of activity, which corresponds to one behavior, the activity levels of individual neurons haven't changed, but the exact timing of their activity has changed. And as a result, we've broken correlations and disrupted normal patterns of coactivity. We have another method, which we call SHART, which stands for Shuffling Activity to Rearrange Correlations. And we actually published this in 2016. And I won't go into the details of this method, but here at each time point, we iteratively reassign which neuron is active in order to achieve target levels of activity and to achieve a target correlation matrix. So here we also shuffle the data, but we end up preserving not only the activity level of each individual neuron, we also end up preserving the correlation matrix if we want to do so. And so now we can train our classifier based on the real data, and then we can test it using either swap shuffle data or shard shuffle data and see if it does better at decoding one or the other. And if it does, it will tell us whether these coactivity patterns are transmitting any additional information above and beyond what's transmitted by activity levels. Okay, so again, we take our real data, okay, we use this for training, and then we split it up. So here's a social epoch, or sorry, this is like a home cage epoch, this is a social epoch, home cage, social. We take each epoch corresponding to one type of behavior, and we either swap shuffle it, which will preserve changes in levels of activity between epochs, but will disrupt changes in coactivity, or we shard shuffle it, which will preserve changes in activity levels of individual neurons and preserve changes in correlations or coactivity. And so when we test our classifier on shard shuffle data sets versus swap shuffle data sets, what we see is that it consistently does better on the shard shuffle data sets. And this is whether we're trying to decode whether the mouse was alone in its home cage or being social, or even whether the mouse was interacting with the first novel mouse or the second novel mouse. In both cases, we see that we're able to decode the shard shuffle data sets better than the swap shuffle data sets. And what this tells us is that coactivity or correlations are transmitting additional information that's above and beyond what's being transmitted just by the changes in activity levels alone, okay? And so then the last thing is we can look at what happens in SHANK3 knockout mice. So we can quantify this difference in performance. How much better is performance for shard versus swap shuffle data sets when we're decoding them after we trained on the real data? And what we see is that this difference, this information that's added by coactivity, is consistently larger in wild type mice than it is in SHANK3 knockout mice. Okay, and this is definitely the case when we're looking at whether a mouse is alone in its home cage or socializing, and it seems like it's also probably the case when we're looking at whether the mouse is interacting with mouse number 1 or mouse number 2. Okay, so why is this, and how does this relate to what I started with? Well, we can actually look at the patterns of coactivity, right? So we can look at 3 neuron combinations that happen when the mouse is either alone in its home cage or when it's socializing, and we can ask, was each 3 neuron combination statistically significant? That is, Do we only see 3 neuron combinations that we would expect to happen by chance, just based off of random overlap between neurons? Or are we seeing patterns of coactivity that are significant and that are unique to this data set and wouldn't be seen if we shuffled the data set randomly? And what we find is that in the knockout mice, during socialization, the fraction of sort of statistically meaningful patterns goes way down. And we think the reason for this is what I started with, is that during socialization, the knockout mice they're recruiting too much activity. And as a result, there's a lot of neurons that randomly overlap in their activity. And this just reflects chance overlap of activity. And this kind of drowns out the statistically meaningful patterns of coactivity. And as a result, our classifiers are no longer able to use coactivity to learn more about social behavior. And so what this tells us is that in SHANK3 knockout mice, because too many neurons are being activated, the way information is being transmitted through the brain is changing. And the ability of coactivity to transmit information has been severely degraded. All right, so I'll just end there and say, in a mouse model of autism, too many neurons are activated. So people often talk about EI imbalance or excessive excitation. And that's what we see. Too many neurons are being activated. And this really is too much of a good thing. People often talk about signal to noise ratio while being a little bit vague about what that means. Here, signal means statistically meaningful patterns of coactivity that transmit behavioral information, and noise is just patterns formed by the chance overlap of activity. And when too many neurons are activated, we get a decrease in the signal to noise ratio. And as a result, the way that downstream neurons can kind of decode behavior changes and it becomes degraded. And so this doesn't just tell us something about autism, it tells us something more general, which again is kind of a fundamental controversy in the field, is how do neurons transmit information? And here we show it's not just the activity levels of individual neurons that all add up, it's actually patterns of coactivity, things you can only see if you look at many neurons at once that synergistically transmit additional information. Okay, so that's it. This is my acknowledgement slide. The first project was led by Kathleen Cho, and we've collaborated with the Rubenstein lab at UCSF and the Spitzer lab at Stanford on this. The second project was led by Nick Ross, called Chart, was developed earlier in the lab by a grad student, Francisco Vilanco. Thank you all.